I want to
thank
thank Tracy for the invitation. It's both for me personally to be a part of this session, but also for just bringing attention to this really important topic. Sex and gender are cross-cutting issues in all of biomedicine, and because they're so cross-cutting, they have really been ignored for many years. But now we're finding, as we've turned the spotlight on sex and gender, the more we look, the more that we're discovering that I think it shocks and amazes. Certainly it does me every day, and I've been doing this for 25 years. So now we're going to turn to some animal models and get into some mechanistic data, but we'll just start with some generalities about how sex differences in the brain are determined. They're multifactorial. Obviously, we start with our genes, as we just heard so beautifully. They're also your environment and your experience, which are going to be hugely important in the human population and impossible to control for. And then lastly, your hormones, as we've also had so nicely introduced this morning by Emil and Tamara. However, in the adult, one of the things people don't realize is that a lot of these sex differences in brain and behavior are actually programmed very early in development. And Neil alluded to this briefly, but gestationally, the male fetus turns on its hypogonadal pituitary axis and produces copious quantities of testosterone, so much so that a newborn baby boy has a circulating testosterone of a 25-year-old adult. This helps frame a sensitive or critical period in development during which the brain sex is designed to match gonadal sex, and as we heard, sometimes that doesn't work out perfectly. In our rodents, this actually extends quite nicely into the postnatal period. In humans, there's a little postnatal, what's called mini-puberty. We don't really understand mini-puberty yet at all, but we think most of the differentiation of the endpoints that we've looked at are occurring in utero. Although you don't know what you don't know, so there's still other possibilities. Fortunately, our rodents clearly keep that sensitive window open postnatally so that we can do something like inject with testosterone or estradiol. As Neil pointed out, it's a masculinizing hormone in our rodents. We can do that to a female and essentially convert her brain male, and that gives us a great hook into the mechanisms by which these processes are occurring at the cellular and molecular level. And so what I'm going to talk about today is just two systems in great detail. I could sit and go around and talk about a whole bunch of different sex differences, but instead we're going to dive down into two brain regions where we know the behavior that they're associated with, and we know that they have existing sex differences. And as I just said, I could just talk about just about anything in the brain. We now know that any endpoint in the brain at some point, in some way, and somehow is going to be different in males and females. We used to think only reproduction was the thing that was different in males and females, and that helps really relegate sex differences research to a backwater. But with the onset of work of people like Bruce McEwen and now many, many others, we know that sex differences are widespread throughout the brain. This is sort of my cartoon version of everything that happens in the brain as it develops. You start with neurulation. Even the formation of the neural tube happens differently in an XX versus XY fetus. We've got neurogenesis, gliogenesis, migration, dendritic growth, myelination, phenotypic differentiation, synaptogenesis, synaptic pruning, and apoptosis. And boom, that's just about it. That's about everything we study as neuroscientists. And I could tell you a just so story of every one of these endpoints. But instead, I'm just going to focus on two. First is synaptogenesis, one fundamental question in neuroscience, how are synapses formed? And second is cell genesis, how are cells born? Also equally fundamental to neuroscience. So the first story I'm going to talk about is in the preoptic area, nothing to do with vision, just happens to be pre to the optic chiasm. And this is sort of ground zero in the brain for the control of male sex behavior. It does a lot of other interesting things, but really central to male sex behavior. If you lesion this brain region, males lose all interest in sex. If you stimulate this brain region, males lose all interest in anything but sex. So it's a nice place to get a neuroanatomical correlation to behavior. And over many years, we have established a really robust sex difference in the synaptic profile of preoptic area neurons. Now this neuron up here in the corner, this is not what POA neurons look like. They're actually kind of skinny, not very attractive. They're not even color-coded, but they are studded with dendritic spines. And what we have found is that the male has about twice the density of dendritic spines as the female, and that means that when he gets a hit of olfactory input from a sexually receptive female, he gets twice as much excitation, and that's absolutely essential to his ability to perform male sex behavior as an adult. So we know that this is established in that critical period that happens early in development. And so we can treat a female with testosterone and we can make these synapses form and say, well, great, here we have a model for synaptogenesis that we can induce with a steroid. What is the mechanism? One would assume it must be a neurotransmitter. That's how synapses are formed, based on neurotransmitters. But the answer is a resounding no. We can go through every single neurotransmitter known to man, and none of them can explain mechanistically this formation of synapses. But instead, it's from a very unexpected player, prostaglandins. Membrane-derived, short-term, short-lived signaling molecules normally associated with inflammation, the thing that gives you a fever, makes you sick, what you take your Aleve and your Tylenol and your ibuprofen for. And what we found in a series of studies done by Stuart Amato, an MD PhD in the lab, was that if he injected prostaglandins into the brain of a newborn female, just like we would of testosterone or estradiol, raised her to adulthood and asks, are you male when it comes to sex behavior, the answer is a resounding yes. She's indistinguishable from a normal male. To ask if she's male, we have to take her ovaries away, give her testosterone, present her with a sexually receptive female, and she will mount and she will act like she has a penis, even though she doesn't. She doesn't seem to know the difference or really care, for that matter, which is interesting to think about. We also found, though, that if you block the COX enzymes with indomethacin, aspirin, nimesulide, anyone you want to use, only during this sensitive period, raise those males to adulthood and say, are you male? The answer is no. It's also, I'm not female either. They simply become asexual. They show absolutely no interest in sex of any kind. So along the way, we found that a single injection of prostaglandins was capable of initiating this process. And that just seemed absolutely crazy. I mean, that seemed like Mother Nature playing with fire. This is a short-lived membrane-derived signaling molecule, and one exposure is going to change the sex of the brain for the entire life? How can this be? And we said, well, we must be lighting a fuse. And I don't know if you can see, that's actually a little mouse lighting a fuse. We must be lighting a fuse that's going to set off some sort of a burning reaction that is going to carry on this differentiation process that's fundamental to all of life. And so we said there has to be a positive feed-forward mechanism, perhaps for the production of more prostaglandins. And so we asked, is there another cell type that might be contributing to the production of prostaglandins? Up to this point, we had also been emphasizing astrocytes quite a bit, but everybody else in neuroscience was discovering microglia. And I said, well, if they don't express GFAP, I'm not interested. Well, I changed my mind right around the time that everybody else was rediscovering microglia. What are microglia? Very poorly named. They are not micro. They are 10% of the cells in your brain, and they are not glia at all. They are actually genuine immune cells, right? They are modified macrophages. They're actually born in the yolk sac when you're little tiny fetus. They migrate into the brain and are rodents at 9.5 days of development. They get in there before the blood-brain barrier closes. It then closes behind them, and there they are, resident for life. And their job is to be the brain's immune system. And what piqued our interest was that studies in injury models had shown that they both respond to and produce prostaglandins. They also, as you can see, take on all these enormous different kinds of morphologies and shapes. They're very facile. And fortunately, their shape is kind of a window into their mood. The more sort of blebby amoeboid-like they are, the more they're going to produce prostaglandins, cytokines, et cetera, all your inflammatory markers. And the more ramified and filigreed lacy that they are, what we now call surveying, the less of these inflammatory markers they're going to produce. So we asked a simple question, is there a sex difference in the morphology of these microglia in the preoptic area early in development? You can see up on the left, there's the male. His microglia show that amoeboid, kind of blebby, angry, prostaglandin, cytokine-producing morphology, whereas the females are very ladylike. They're nice, they're sitting there. They are also surveying, though, so they're busybodies. They might look good, but they're checking on everybody around them. That is their job. And we went on to show that those male microglia are, in fact, producing more prostaglandins than the female microglia. But how do we get from prostaglandins and microglia to dendritic spines? Well, I could go on and present a whole lot more experiments, but instead I'm just going to summarize it all with one cartoon here. This is with apologies to all the students whose entire thesis is being reduced to one little part of this cartoon. But as you can see, we have a big yellow neuron on the left, we have an astrocyte, and in between a microglia in the corner, and a dendritic process over to the right. So we're going to start with testosterone. It's going to fall out of the sky and be aromatized into estradiol, where it's going to bind to the estrogen receptor, translocate to the nucleus, and transcribe the COX-1 and COX-2 enzymes, converting arachidonic acid into PGE2, which is going to bind to the EP2 and 4 receptors on the dendrite, which are adenylate cyclase linked, making cyclic AMP, phosphorylating PKA, activating it to phosphorylate the GLUR2 subunit of the AMPA receptor, causing them to cluster at the membrane. Prostaglandin is also going to cause the microglia to make even more prostaglandin, and this prostaglandin storm is going to cause calcium-dependent glutamate release by the astrocyte, which is going to bind to and activate the AMPA receptors, causing the formation and stabilization of dendritic spines. Thank you. Thank you very much. I'll be here all week. All right. Now, you might have noticed there's another cell type that was lurking in this diagram, and that's because I made this good diagram quite a while ago, but we were starting to get hints that there was yet another player in this interesting story, and that was the mast cell. And my lab was like, what the heck are mast cells? Mast cells are the other immune cell of the brain, but they are an immune cell of your entire body. They have a completely different origin in your microglia. They come from the bone marrow. They're from a completely different lineage. They are everywhere, and they're mostly on the surfaces. They're on your skin, your eyes, your nose, the lining of your mouth, and they are what gives you an itchy eyes, a runny nose, a rash, and also anaphylactic shock. So they will annoy you and they will kill you. So they're the kind of considered the first responders in any kind of injury reaction. So we asked a simple question: is there a sex difference in mast cells in the neonatal POA, the exact same time that we're looking at that sex difference for the microglia? And here's what mast cells look like. They look like dirt on your slide. So you can see there's the center there is the third ventricle, and down to the bottom you can see some little black spots that look like dirt. Those are actually toluidine blue stained mast cells. This is how you count mast cells. 1, 2, 3, 4, 5. I think there's 6. Yep, there's 6. I'm done. Okay, let's go to lunch. Everybody in my lab wants to study mast cells now because that's how difficult it is to count them. And so we did an exhaustive survey of mast cells across development. And you can see here across the first week of life, the blue, I use the traditional approach of blue is male, red is female. You can see that the males have substantially more mast cells than the females. It goes away right at the end of the sensitive period, goes way down low, little bump up in adulthood, but there's no sex difference. I also want you to note, these mast cells are deep in the neuropill, right? That is not where they belong. Mast cells are traditionally found on surfaces, so they normally hang out in the meninges. This is the only region of the brain where we found them deep in the neuropill, and they were only there for the first week of life. So fortunately, because mast cells will both annoy you and kill you, there are good tools to study them in terms of you can stop them from degranulating, which is life-saving, or you can induce them to degranulate in order to study their function. So we took the approach then of just manipulating the mast cells, nothing else, pharmacologically manipulating the mast cells to either stay calm or dump their contents. And then we asked the question, are they talking to the microglia? Because that's what has been known in the injury world. This is the way this relationship had always been studied. There's a penetration of the blood-brain barrier. There's a bunch of signals released. The mast cells run to the scene. Woo, 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 woo. They dump their contents, boom, and that calls in the microglia, and the microglia come running in, and they come and they clean up all the dead bodies, put them, throw them away, say, okay, everybody, nothing to see here. I'll go home now. It's all over. So is this going on in the developing male brain? Basically, is there a train wreck going on in the male brain? And so what you can see is on the top is the normal, what I showed you, that normal sex difference. Down here on the left-hand corner, that's a male whose mast cells we just told, calm down. Just calm down and stop degranulating for 2 days. And you can see that his microglia are absolutely indistinguishable from the female, which tells us something very important. His mast cells are constitutively degranulating, right? They're just sitting there continuously degranulating and refilling over and over and over again. The far right lower corner is the female whose mast cells we just told to degranulate for 2 days. And her microglia are all blebby and all worked up and indistinguishable from the male. These are the actual bar graphs, so you know what they really look like, so you know I'm not just making that up with my representative images. And you can see we've done a complete sex reversal of the microglia morphology. Big question is, what language are those mast cells speaking? They dump all sorts of things in their contents. To actually find that out, you can't, there's no microdialysis probe, you know, there's nothing we can measure here. We're counting these cells in the tens, right? These are exceedingly rare events. So you have to take them out and culture them. We get a pure mast cell culture. It takes weeks and weeks because we've got to throw everybody else out. Treat them with estradiol and found out that they are in fact mostly dumping histamine. So then we take that cell culture and we ask, what is that histamine doing? We have to do this again in culture, and we did a mixed neuronal astrocyte microglial culture, and what we found is that the histamine is promoting prostaglandin production by the microglia. If the microglia aren't there, we don't get any prostaglandin. So they are absolutely, there's a clear handoff from the mast cells to the microglia. And then lastly, sort of the money experiment, if we just manipulate those mast cells in the first couple days of life, raise the animals to adulthood, we have masculinized the females in terms of their male sex behavior. I'm not saying there's anything abnormal here or anything. I think this is natural variation. This is not a disease model. This is just manipulating the normal process of sex differentiation of the brain. The important point, though, is that to make a male brain, it actually takes 4 cell types. 2 of those of neuronal origin, the neurons and the astrocytes, but 2 of them are from the immune system. And we can't get to the male brain without the immune system. All right, now shifting gears, we just took 2 steps to the left and went to the amygdala and wanted to study another sexually dimorphic behavior, which is play. Juvenile play is complex, it's robust, it's multifactorial, it's a reciprocal between the 2 animals, and you can count all these different kinds of components of it. But why, again, would you study play? Well, first of all, it occurs during a restricted developmental period. It's a time when there are no steroids around. It's more physical and intense in males. We call it rough and tumble play, just because it is more physical and intense. The sex difference is established early, during the same time that the sex difference in mating behavior is established, and it has strong face validity to humans. And in fact, across all mammals in which play occurs, males routinely play more intensely and physically than females. The circuitry of play has been worked out for the most part. It is, not surprisingly, largely overlapping with the social circuitry of adulthood that we're so interested in. But one interesting aspect of it is that people used to study this a lot. You don't hear as much about play anymore. And I'd say this is ancient history because this was Michael Meany and Bruce McEwen, yes, that Michael Meany and that Bruce McEwen, who showed in 1983 that testosterone was a masculinizing hormone when it came to play, and it did so through the amygdala. Now, we had seen a sex difference in cell genesis in the amygdala based on BRDU dating. And it was an unusual one because we found the females had more BRDU positive cells than males. And this was a surprise because we'd found that males had more BRDU positive cells than females in the hippocampus. So we were all excited that this was finally something that was more in females, and it was interesting. But we didn't have any idea of what the origin of this sex difference was. Well, at this point, I couldn't get anybody in my lab to work on anything other than microglia, so we returned to the microglia, which we know can both promote and inhibit cell genesis. And we saw the morphology of the microglia before as being quite complex, but it's even more complex than we thought, which, as you can see on this microglia here, are these giant phagocytic cups. In the study in which we'd done in the POA, when these cups, we finally started seeing them, and the postdoc says, yeah, I saw those too, but I didn't know what they were, so I just ignored them. Oh my God, how can you ignore those? Look at those things, they're giant. So we've had great fun imaging these microglia. This is, I call my Baltimore lacrosse microglia. Appropriate here to Florida, on the left we have the Mickey Mouse microglia, on the right is the double D cup microglia. And so we just asked a simple question: is there a sex difference in this phagocytosis by these microglia during the sensitive period for sex differentiation of the brain? And here you're seeing across the first week of life, blue males, red females, and you can see that the males consistently have more phagocytic microglia than the females. Also note the rate. This is the percentage of phagocytosis. Up to almost 30% of these microglia are phagocytic at any one moment in these first few days of life. This is a process that's estimated to take 20 minutes to 2 hours, and when we're taking a snapshot in time, we're finding 30% of them are engaged in phagocytosis. So that's a pretty impressive amount of eating that's going on. Again, to remind you that this process all occurs during the sensitive period, and we can convert females to males by treating them with testosterone or estradiol. So we did exactly that. We treated the females with testosterone. Here's the sex difference: the female on the left, the male on the far right, and the female treated with testosterone comes exactly up to the male level when we treat her with a dose of testosterone. And that's perfectly inversely related to the number of BRDU cells. So we have a nice inverse correlation between phagocytosis and the number number of newborn cells. So that leads to the obvious question, what are the microglia eating? We were pretty sure it was cells based on the size of those phagocytic cups, which are on average 8 to 10 microns, some of them as big as 15 microns. So this is not nibbling, this is not Beth Stevens synaptic pruning, this is gorging, right? They are eating full-blown cells. So 80% of those cups contain DNA material, but the real question is, are they eating newborn cells? Is this a preferential diet for the young, right? It wants to eat only newborn cells. So we used PCNA, a proliferating cell nuclear antigen. Cells express this protein for about 48 hours after they've been born. This is a microglia. The PCNA is in red. You can see that dendritic cup down there with the PCNA. And I will say, this is the pattern we see over and over again, that the microglia just shoots this arm out and grabs a distant cell. It doesn't seem to eat its immediate neighbors. They always seem to reach out for some reason we don't know. But you'll also note that this microglia itself is expressing PCNA. It was just born within the last 48 hours. They don't have to take time to differentiate like neurons and astrocytes, and they are born hungry. Here's another image of one as our imaging has improved. You can see this one is actually turning back on around itself. It's a particularly juicy newborn cell that it had to have. And if we look at that close up, oh, shoot, oh, there it is, okay. You can see that the nucleus of this PCNA positive cell is fully intact, right? Suggesting that that was quite a healthy cell that was suggesting. When we quantified this, we found that up to 60% of these cups contained PCNA, suggesting that they are preferentially eating the newborn cells. But what about dying cells? I mean, that is what they're supposed to be eating. And we did then look at cleaved caspase 3. Here you can see the cleaved caspase 3 is in red, a giant phagocytic cup eating a dying cell. And you'll note there that the nucleus is not healthy. The nucleus is beginning to show signs of apoptosis as the DNA is breaking down. When we quantified that, only 10 to 15% of these phagocytic cups contain dying cells. So this suggests that we're looking at a different phenomenon than just classic phagocytosis. It's in fact phagoptosis, as named by Guy Brown and Jonathan Nahar, Jonas Nahar, which is that they combine the terms phagocytosis and apoptosis. And what it is it's the consumption of a living cell. So cells, when they're in distress, will sometimes flip out an eat me signal because they're like, oh, life is too hard, I can't go on, I want to die. And so they put out this eat me signal, and if nobody comes along and eats them, they go, oh, actually, maybe it's not so bad, you know, I think I've changed my mind, I think I would like to live. And they can put the eat me signal back inside their cell, and they can go on and live a long and healthy life. So there's a whole bunch of different kinds of eat me signals. We focused in particular on the complement system for background reasons I won't go into. They also have don't eat me signals, which is an important balance between the two. But we asked the question, if we just selectively inhibit the phagocytosis by blocking that eat me signal, what will happen? So to do that, we injected a blocking antibody to CD11B, which is part of the C3R complement complex, and we put that in one side of the amygdala and vehicle in the other. And the prediction would be that in the side that had the vehicle, phagocytosis would occur, and you'd have this number of BRDU plus cells. But the side that had the antibody, if those cells were in fact viable, they would now survive, and you would have more BRDU positive cells. That is exactly what we saw in the males. So you're looking at the left and the right hemispheres here, the vehicle versus the antibody, and the number of BRDU cells increased in the males that were treated with the anti-CD11B antibody, not in the females. In the interest of time, I'm not going to go into why that difference occurs, but we do know if somebody wants to ask later. So how do we collect microglia phagocytosis to sex differences in play? We repeated that exact same treatment. Now, this treatment's being done in the first couple days, and we now raise them to the juvenile age, and we ask them to play. You see the nice sex difference here, female on the left, male in the middle. He plays more, unless he had anti-CD11B, at which point his play is at the level of females. We then looked at whether or not that was impacting neuronal excitation during play. We used ZIF268, an immediate early gene, and we can see that naturally in males and females, the neurons are more excited in the males, and if they had their phagocytosis blocked when they were babies, their neuronal excitation drops just in perfect alignment with their play. So what were these newborn cells destined to become before they were so maliciously engulfed by those phagocytosis? There's a number of options. They could be neurons, they could be astrocytes, they could be oligodendrocytes, they could be microglia, they could be all of the above. To do this, we injected BRDU in neonates, and now we waited 3 weeks to give those cells full time to become mature, reach their full identity, find their inner selves. And not surprisingly, given the pattern of BRDU, which we had seen, which was rather uniform throughout the amygdala, not a neurogenic zone, we found that 80% of them were destined to become astrocytes, 13 were going to be neurons, and 7 were going to be microglia. And no, we did not fudge those numbers. Those were done completely independently, so we've accounted for 100% of the cells, but most of them were going to be astrocytes. We then went back and used an astrocyte precursor marker and showed that the males were actually eating more astrocytes than the females. You can see one here. He's got an astrocyte going down his gullet, glug, 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 as he consumes it. And this results in more astrocytes in the female amygdala than the male. So female on the left, male in the middle, and if you had the anti-CD11B, you increase the astrocyte population in the males, which reduces their neuronal excitation during play, which reduces their play. So when we think about all of the things that can be affected in the brain, and we thought we were studying a neurocell genesis phenomenon, we were completely wrong. It was actually murder. So the conclusions are that microglia are critical purveyors establishing sex differences in the developing brain. The mechanisms are region-specific. There are other brain regions with other things going on, as are the impacts on the neuronal and astrocytic population. And this results in enduring changes in brain and behavior. And there's many things that I've left out, but 2 are important. One is I didn't tell you the mechanism by which the microglia are inducing the phagocytosis. That has to do with endocannabinoids, which are higher in males than females, the endocannabinoid tone, the anandamide. And second is, why the heck are males more inflamed than females? Where does that come from? Testosterone is actually anti-inflammatory. And we're working on the hypothesis that it's all mom's fault. It starts with the maternal immune system attacking male fetuses preferentially over females. Thank you to the funding agencies who have supported the work, and to all of the wonderful students and postdocs who have done it. Everybody in red contributed data directly to this talk. Thank you.
Thank you, Dr. McCarthy, for that fabulous talk. We have time for a couple quick questions. You've just wowed them, and oh, here we go. And can you please repeat the question?
Thanks for a fabulous talk.
I
was just wondering what evidence you'd found towards the idea that some of the hormonal influences might be dependent on the sex chromosome context, and some independent. What sort of evidence is there for those 2 possibilities?
Okay, so Armin asked basically what's the role of sex chromosomes, coming from a man who studies sex chromosomes, and how is that interacting with the hormones? And what we hypothesize, and we only have little tiny hints of tantalizing data so far, is that the genes on the Y chromosome, such as that make the HY antigen, or Norexin 4Y, or major histocompatibility complement proteins, are actually being recognized by the maternal immune system, and that's causing an inflammatory response in the male brain, either by indirectly through like prostaglandins and cytokines, or possibly through direct invasion of maternal immune cells, such as mast cells, into the fetal brain. And so the way to find that out is to take a female and give her a male immune system and then get her pregnant, and you would predict that there would be improper masculinization. So we're really in the thick of that right now. Turns out doing a bone marrow transplant is easy, to give a female a male immune system, but getting her pregnant and getting her to deliver after that is not. So that's where we're at.
Thank you for making science fun. First, I'm curious about it was a lot of fun. So I'm very curious because a hungry astrocyte has a dysfunctional energetic system, because it needs more energy for producing the cells. Have you checked whether on the process of that engulfment of more the new cells involve a dysfunctional mitochondria? If you stabilize your energy system, can you restore, maybe engulfing more the dead cells and not the new cells?
That's a very interesting idea. So her question was because of the importance of energy regulation in astrocytes, whether or not there's mitochondrial dysfunction that is contributing to those precursor cells perhaps putting out their eat me signal. You might say, and I didn't show you data that these newborn cells do have a higher concentration of C1qa and C3b, which are both prominent eat me signals. That's a very interesting idea. We hadn't thought about that because, as I mentioned, we are focused on the mechanism is actually endocannabinoids, which I didn't show in the interest of time, which we think make the microglia move more in the male, and they just have a higher mobility and motility, and they just encounter the newborn cells more frequently. But it's a very interesting idea, and one worth considering because there are even sex differences in mitochondria.
Exactly.
So, yeah, thank you.
Cannabinoid specifics too.
Yeah.
Excellent.